Today's speaker is Dr. Jinhua Wang, who's an associate professor at NYU School of Medicine and a member of the Genomics Center at the NYU Cancer Institute. He has done lots of pioneering work in genome sequencing, and he's speaking about sequencing depth and coverage today.
Thank you for coming. It's a great pleasure to talk today. So coverage sequencing coverage is, I think, very important to design the experiments. So when we can, give an experiment, we want to know how many runs, how many samples we need to get sufficient data. Also, it will, actually affect how we understand the results or how we interpret the results, given the coverage and given the depths, and how to estimate the false discoveries, including false positives and false negatives. So this is a standard genomic view realization of a sequence project using any genomic viewer. You input your sequence alignment, you will see the reads aligned along the chromosome. So the depth refers to, for each position, for each nucleic acids, how many times it appears in the reads. And so sometimes it's interchangeable with the coverage. So, but the coverage also refers to how many regions how much the proportion of the genomic region that you capture, that you have information, that you have data that you can work on. So the discussion, the study on coverage actually goes back to 1988. Doctors Eric Lander and Dr. Michael Waterman. So this famous Smith-Waterman alignment algorithm, one of the authors, they actually started discussion on this. So this is the first publication titled Mathematical Analysis of Random Clone Fingerprinting. So when they investigate the problem of physical mapping to investigate the relationship of the number of clones, the length of the clone, and how many contigs, how many islands, so how many samples you will get, basically investigate the relations. And then this coverage, so they first used a notation of coverage, in this form. Basic coverage is the length of the reads multiplied by the number of sequence and divided by the genome size. So in this, using this formula, actually there is some estimations on, to give a genome, let's say 1x coverage or 30x coverage for different platform using with different lengths of the reads, how many, roughly estimation, how many reads do you need? So this is actually published in a review paper on Nature Genetics. So this is a survey of the coverage of current sequencing projects. So this survey looked at about 800 studies, including whole exome sequencing, whole genome sequencing, ChIP-seq, and RNA-seq projects. So in this figure, to generate this, actually, they summarize about 800 experiments, the number of reads that they use in the study. And so basically what we see here is actually for exome sequencing, it's a kind of a multi-model, so that due to the different targeted depths of studies. And for RNA-seq, we see a wider range span, multi orders that shows the the coverage for RNA-seq, really varies because of the expression levels difference. So following, I will discuss the coverage-related issues for whole genome sequencing, exome sequencing, RNA-seq, and ChIP-seq in this order. The first human, this is actually the first reference human genome. So now we use a lot of the reference genome, whatever project we have, be it exome sequencing, resequencing, targeted resequencing, exome, transcriptome profiling, ChIP-seq study, we all will use this reference genome. So this first reference genome published, I think, more than maybe 10 years ago. So the depth of the genome, the draft genome is actually 4.5x coverage. And the finished genome is kind of 8 to 12x coverage. So that's we see a continuous updating of genomic version of genome versions. From HG probably 16, 17, 18, 19. Still keep updating. So the draft genome, the coverage is very low, and then because of this repetitive regions that are hard to access, some particular region that is not sequenced sufficiently. So this finishing actually adds more and more risk to achieve a better quality. So that's why when you use HG18 or HG19, different human genome versions, you will see many actually different annotations, different structure. That's not a surprise because they really keep updating, adding more reads, and changing the assembly. So the low coverage here doesn't mean the quality is not good. Comparing to here, we are talking about 30x, 80, 100x. Oftentimes, the low coverage here doesn't necessarily mean low quality, because the major factors that determines the requirements of depth depending on the sequencing technologies, the error rate of the platform. So this early they used Sanger sequencing, so the error rate is very not relatively low compared to the short read sequencing, and the read length is longer. So when you assemble the genome, you don't need that high depths. So then there are a few other whole genome sequencing, like for this the panda genome. After that, there are a few other whole organism sequencing. The panda genome actually achieved shotgun sequencing 70x coverage. But the quality means the number of longer contigs, the number of gaps is not as good as the lower coverage sequencing using Sanger sequencing. At the same time, the dog genome, I think probably about the same time finished. It get about coverage of 7 to 8-fold. But still have achieved a better quality than the panda genome. So after the reference genome was done, this 1,000 genome project. So when we do variant calling, variant study, we refer a lot to the 1,000 genome data. When we do annotation, we will filter, mutations that we see in the 1,000 genome, and we utilize a lot of the 1,000 genome results. The coverage for the 1,000 genome is actually very. There are 3 layers of data. So the first layer is the trial sequencing. The trial is actually a whole family, the father, mother, and the child. So this sequence both the parent and the son and daughter, and have very deep coverage, like 40x coverage. And there's also low coverage whole genome sequencing. The low coverage is about actually 2 to 6x coverage on different populations, like 60 European, 60 Central Europe people, 30 Han Chinese, and 30 Japanese people. So this is about 200 samples. Other individuals, they actually sequenced the exon sequencing. The coverage reached about 50x coverage. So from this three group, there actually we get lots of SNPs, but the quality of the SNP is we really should consider that when we use this data. So the following study shows the false discovery rate for the trio is relatively low, like at 2.3%. For the low coverage, it's about 10%. And for the exon, it's relatively close to the trio, the false discovery rate. Also, for the number of mutations that discovered from different set of data, we see unsurprising, we see a lot in the low coverage sequencing data set. So when we have this in mind, when we use the data, we have to actually consider how reliable is the SNP call from the southern genome. So when we filter our variant call from individual sample, from our patients, we need to definitely take this into consideration when we refer to some public data set. Good news is actually, I think there are different groups actually are trying to improve and also include more samples into the for variant call to try to build a better data set to compare. So this cartoon show for the different data sets. For the trio, the coverage is really low, really high. And the mutation, the SNPs actually are phased. The haplotype of the SNPs. So which SNPs comes from which parents. And for the low coverage, the 2 to 7/6 coverage, those about 200 samples, the coverage is really low. For the SNPs, the phasing is actually is using some statistical models, and haplotype blocks relatively is smaller. For the exon data, for the left data, the SNPs from exon sequencing, they are totally unphased, but the quality relatively is better than the low coverage SNP calls. So this is for the whole genome sequencing. So we understand, different coverage will produce different quality variant calls, and some low coverage project may sometime actually produce maybe compared to high coverage data set. Maybe it's better depending on the platform. Also the quality is affected by the algorithms, the sampling algorithm, the alignment algorithm. So following, I will talk about the exon sequencing. So for exon sequencing, the capturing, so there's lots of techniques, actually, different keys to capture the exon, the coding part. And here is actually show the correlation. This is actually a figure from one exon sequencing provider. So this shows the correlation between the the percentage of targeted regions covered and the sequencing depths in the human exome sequencing. So as an example, if the coverage is 30x, so basically you will see actually 50% of the less than about 50% of the regions is covered less than 30x, and if you want to achieve higher percentage that covered by 30x, you may actually need 100x coverage. Also, the SNPs discovered actually goes you have high coverage, you get more SNPs. That's not a surprise. But at 200x, you still don't see it goes to a plateau. So you will see the increase. You discover more mutations as you increase the coverage. So there's various factors that will affect the mutation call. Coverage, sequencing depth is one of the factors. So this is one patient data that we analyzed. And we have the exome sequencing for this patient. And we have 3 methods to call the mutations. So the results shown here actually tells us, there are different methods really capturing different sets of mutations. So there are many, factors that can affect, this kind of discordance. Why some algorithms call the mutations, but the other will miss. The assumptions of different algorithms and the filtering that different algorithms use, how they deal with the depth, how they deal with the coverage, that will all affect the output, the final call results. And from here we can see for indels, there's a much less overlap for indel calls. That probably is due to not sufficient coverage. There is a study actually systematically look at the exome capture, exome sequencing using different capturing kits, and to investigate how many reads is sufficient to really covers to call all the mutations in the individual. And here we see this solid line are the for the different platform specific mutations, the solid line. And the dashed line is actually the total number of mutation codes using different exome capturing kits. So here what we see is actually the trend that the number of mutations that you will discover really goes with the number of reads, but at for 30-40 million reads, that region, it seems like the curve is going kind of flat. And also for different annotations, like for when you call the mutation, you align to the genome and then you refer to the gene annotation. So for different gene annotation, it also behaves a little bit different. For this CCDS, this is the common consensus gene. That's kind of an annotation, or a way to get this annotation. And also for REF-seq, the curves show a little bit different. So the big difference here is actually on the UTR region. So we are interested in, for us, we are interested in looking at regulatory mutations. So you can imagine, the exome sequencing covers when you look at you get your alignment, you will see some of the mutations, some of the regions actually is in the UTR region. The capturing, the coverage and how many regions you will see in the UTR is actually, depends on the platform. But we do get the region that covered by the reads, so we can call the mutations in the UTR region. So here the pink line shows the shared mutation caused by all these different technologies. So the trend of this line shows that at the data they evaluate like 50 million reads, you still see it's kind of keep up going. So different technologies really capturing different set of mutations. And I think the current coverage for one single platform, if you at a certain coverage, some mutations some reason you cannot capture. So that's a coverage issues. So the different capturing technologies will have their regions that the other will not capture. This is for indels. Basically, for indels, the indel code compared to single mutations is actually can be more accurate. So for the variant code, the reads compared to mutations, somatic mutations, you need more reads. So at this level of 50 million reads, the number will keep going. So with mutation frequency, we can do lots of things. But like for cancer, for tumor sample study, the tumor heterogeneity, basically when you look at the frequency of the mutation encoded for the driver genes, you can actually deduce how many clones, subclones in the tumor samples. So this heavily depends actually on the coverage. The more coverage and the more very rare mutation you can discover, the more accurate you will deduce the clonal composition of the tumor samples. Here is actually one of our studies to look at the tumor cell lines that, a mixture of different tumor cell lines using mutation frequency, try to deduce how many cell lines we mix together. So this kind of study heavily depends on mutation frequency, which is actually affected by the coverage, so by all different mutation core methods. So the coverage will actually play some roles in this kind of analysis. So I will discuss a little bit about the very rare mutations. The current whole exome sequencing that is kind of limited by the error rate of the platform. Also the error rate of the platform, also the calling algorithm. So it's difficult to discover very rare low frequency mutations using traditional methods. So for very rare mutations, if it depends on the technology you use, and also depends on the coverage. So if we fix the technology accuracy and then evaluate how many coverage we need to discover the power that you can discover very rare mutations. So we see this different curve that shows that we can actually discover very rare mutations at a reasonable power at as low as 0.1% of the mutations. So this is for exome sequencing. So we use a lot of the next-gen sequencing for transcriptome profiling. So for transcriptome profiling, because of the complexity of the transcriptome, it's a really, I mean, the things you discover, the coverage you need really depends on the biology problem, what you want to discover. So this picture shows the complexity of the things can be transcribed from the genome. So normally we have this coding genes, but there are many non-coding short regulatory RNA transcribed from these enhancer regions, promoter regions, these short non-coding RNAs, and also can in the transcribe can come from this non-coding intronic regions, these intergenic regions, the overlapping gene regions, and some Addison's transcripts. So this complexity of transcriptome, some of those things expressed very low. So we can, when you want to discover that, it definitely needs very high coverage. So this is a simple cartoon to show the composition of the transcriptome. Some highly expressed genes from highly expressed genes to low transcribed units. So when you change the number of reads to cover number of reads that you can get from the transcriptome, so you will see some difference. So this is just to show the the cartoon, but it doesn't carry any more meanings. So in the paper in 2012, there's a very systematic study to look at the expression range, dynamic range of the coding genes, non-coding genes, and novel intergenic genes. So this is the contour plot. So in general, we see that the coding gene has relatively high expression levels, but this intergenic non-coding has a low level expression. So we are investigating what genomic signatures, genomic features can be associated with that. But this figure just to show the complexity of transcriptome, that to address that, the sequencing project, profiling project needs really consider very carefully about how many reads, how many sequence we need. Besides the wide range of expression profile, the splicing form of the transcript also make this transcriptome profiling more difficult. So this is the simple cartoon of the splicing, different splicing forms. You have alternative promoters, alternative polyadenosine sites, alternative splicing sites, skipping exons, exon cassettes, and the mutually exclusive exons, intron can be returned in the transcript. So and for one single gene, you can have actually any combination of this, So make it very sophisticated once you get the reads, which isoform is used, how to deduce the isoform information from the read counts, from the read alignment. So this is actually a summary of the number of annotated exons per gene, and also the number of expressed isoform per gene. So this estimate number exon per gene is from the genome annotation, from the alignment of ESD sequences, from all different computational resources. You can have an estimated number of isoform, but not all the isoform is expressed. So if from it's reasonable, so for genes with less number of predicted isoforms, the expressed number per gene is also low. But for the high number of for the genes with a high number of isoforms, you will see it's not all of them are expressed. Some of them are not expressed. So this tells us that story. Also, the number of annotated isoform per gene, we will see that the major isoform you know, if a gene has multiple isoforms, some isoform will express at higher level, some express at a lower level. So you call it a major isoform or a minor isoform. So if a gene has more annotated isoforms, its major form, you know, will have a low expression from the statistical. You know, you can see it goes down like this. So how many? So I mean, when we understand this, you know, complicated nature of transcriptome, so it's not a surprise to see for transcriptome, you probably need much more reads to really capture the whole spectrum and to capture all the alternative isoforms, the differential expressed isoforms. So in this study, they actually investigated the number of reads that you need to achieve a better performance. So this figure is actually very informative to see, you know, the correlation. So if you have, let's say you have a study, you have, you know, a number of, let's say 500 million reads, and you sample like 300 million reads, and you see for each condition how the correlation, you know, if you discover the same results or not, you know, from different sampling, different simulations. So from here we can see for differential expressed genes, the ideal coverage would be like in this region. So you will see the correlation really goes to the, you know, high level, like 200, 300 million reads. To identify isoform, to identify alternative isoform, differential isoform, you need actually more reads than to find differential expressed genes to discover, you know, differential alternative expressed genes. That's I think it's, you know, it's kind of natural, because isoform, when you deduce that, you know, it's more sophisticated to deduce the structure, to deduce the combination of different isoforms. So finally, I will discuss the ChIP-seq, you know, the location analysis. So there, you know, by location, analysis, I mean, there are many experiments try to map some interesting interactions between DNA and protein, between, you know, DNA and DNA, DNA and RNA, you know, the interactions. Basically, the idea is to capture those interesting regions and get them sequenced, and then map the sequence back to the genome to see where the signals are, where the peaks are. So how many coverage, how many reads we need for that? That is actually really changing between you know, for different scenario. You know, it depends on the things you investigate. So here I will have some focus on the ChIP-seq. So for ChIP-seq, how many reads you will need really depends on the you know, the factors, the point sources, the factors. So there are three types of factors actually. What they you know, define as point source, broad source, and mixed sources. So point source are those, you know, like transcription factors, highly localized factors, or some, you know, histone modification that highly localized and very close to, you know, transcription start sites or promoter regions. So the broad source are actually those, you know, histone modification markers. There are other mixed source that actually it's like the factors like you know, RNA polymerase II, you know, different. This will produce some sort of peaks, you know, in this form. And to actually this review paper in Nature Genetics summarize all this, you know, location study, come up with some rough estimation of how how many reads do you need actually for different type of analysis for the location study. For ChIP-seq, I feel like this is really, this bigger, higher than 10 million reads for, point source for transcription factors, and higher than 20 million reads for other broader source factors are on the low end estimation. So there's very few systematic study on actually to look at how many reads do you need. So this study. Okay, when we align the reads from differential marks, we can see this is actually H3K4 modification. This is for H3K27 trimethylation. So for this factor, this is like a point source factor. It's kind of localized, co-localized with transcription star sites or with promoter regions. So you see with increased number of reads, the peak distribution stays almost the same. There's not much change. But for this H3K27, you do see, with increased number of reads, you see the profile, the peaks under, the annotated peaks or the predicted peaks, are different. So the same metric to evaluate how much reads, you need. For different factors, using this simulation to see the correlation coefficient, you will see actually for H3K4, H3K36 they behave the same. So 20, 40 reads maybe is sufficient, but for H3K27, you definitely need more reads to achieve a better results. So in this study by Peter Park from Harvard University, they actually published this in Nucleic Acid Research, investigating different factors, and we will see actually for different factors you do will consider different coverage, different number of reads. So when we do the simulation, you generate the curve for the reads increased number of reads and the correlation coefficient. You can use that type of curve to do this theoretical prediction to see how many reads do you need for certain experiments. So for this one, basically you want to see if you increase the number of reads and the gain of coverage is less than 0.01, you will call it a sufficient coverage, a sufficient number of reads that you need. So use this kind of analysis, so they predicted the sufficient depths for different markers, and actually this is the results that this paper published. So this is all I have to say for today. So I think maybe some ideas still is not very clear to you, but definitely we can have follow-up discussions, very detailed discussions on this. So if you have any questions, please raise your hand. Yeah.
So you mentioned two slides back that there may be intron retaining in the RNA as one variant.
Yeah.
Right? But if one were to discover a new intron retaining species, you'd have to use reference genome that's both RNA reference genome and DNA reference. Is that right? And is this done routinely?
Yes. So this is basically based on the annotation of the gene.
Right. But it's not annotated, and
If it's not annotated,
you would never discover it, right? Unless you reference back to.
Yes, correct. So that's why there's some ways to mitigate this. There are different predictions. There's different annotation sets of the genes. Like here I showed the ref-seq, like CCDS, like ensemble. Ensemble, probably the most comprehensive. They include lots of computational predicted genes. And they actually also integrate lots of like ESD data into their prediction. So they have the more comprehensive data set. This is the one thing. So basically, we try to explore the whole collection of annotations that we can utilize. And the second thing is there are some new ways to analyze the the RNA-seq alignment data. Just look at the coverage, not look at the read counts or where the reads, but look at the coverage to try to deduce some signals from that. So this is some latest developments, but I'm not sure how good that algorithm perform.
Right, because I actually, this was actually reported, so I knew to ask to look against DNA genome after I did RNA-seq. So it turns out there's a new RNA species that was reported that actually is misspliced. So RNA runs into intron and has a different 3' UTR. So there's that 3' UTR sequence that's there in the sample, but it was not picked up by reference genome because it had not been annotated. But I think I guess one word to discover new species.
Yeah. There's, I think there's when we analyze the coding capacity or the base pair composition at these regions, you will see some different signals. If this is the UTR region, you see, we see some, this region, but it's not annotated. But the signal can be picked up by the sequence content or some signals. Some signals, I mean, like, the TSS star side signals, some, splice signal, GT, EC, this kind of analysis. So we can try other ways to rescue, this kind of information.
Can I add a comment on the code question? We usually deal with this type of problem separately. So we deal with the gene expression profile based on the existing location, and also if purpose of the study is focusing on a new RNA species or alternative splicing, we will basically do de novo transcriptome assembly. That will allow us to discover those new RNA species. I would say it probably a good strategy for this type of study. Really hard to mix these two types of studies, transcriptome profiling and the new transcripts identification together. If we do that separately, we will be able to.
Right, I forgot to mention this de novo assembly of a transcriptome. So for that, also there is this coverage issues. So for de novo assembly of a transcriptome, you need much higher, coverage, much more reads to really assemble accurate, giving this very short reads. Maybe parent reads may be better, but still the coverage is a big issue for de novo assembly of transcriptome. Another question is for de novo assembly. Once you assemble the transcriptome, you need some computation to do this coding capacity prediction. Still, there is a lot of computational work to do. As I just mentioned, to evaluate, whether it is non-coding, what type of new RNA species is that. So yeah, I agree. De novo is the way that can provide some additional information on that. Yeah.
But I guess there has to be a lot of data. I mean, this is going to be, the data will be built, but these low-abundance transcripts of these, apparently spliced transcripts are probably tissue specific, or
yes.
specific, and it's going to be, require a lot of allocation from all sorts of different.
Right, exactly.
So I have a question regarding the variance identification and mutation coding. Based on your experience, what protocols or software do you recommend for identifying these variants and so on to get the balance between the sensitivity and the specificity?
This is a good question. So, GATK, they developed this. Actually, they first developed this for SNP calls when the 1000 genome gets a lot of sequences, low coverage sequences. So they developed this method, try to integrate utilizing the haplotype information, the SNP information into their call methods. So you have to build a model, not you have to, but the algorithm will run this using the data you provide and then give some calls. But I think the development of this GATK also provide a solution for de novo mutation call. That may be, this method, I think there is a lot of the huge user base use this method. But what I want to say is that for individual genome, individual sample mutation call, it's really a matter of, you first look at the quality of the data, the alignment, the overall coverage, and some kind of filters you will design to fit these specific samples. Like for tumor samples, you have different. Some tumors have lots of copy number changes, lots of copy variations. Some very large scale structural variations. So integrate that, the call method will be different. So, but my suggestion is the SAM tools, all these traditional tools that you can look at alignments, and also some like what do you call that? Somatic sniper. This the different methods. I just showed 3 or 4 methods. You look at different methods. Try to see which actually which methods have the best calls. Because different methods, they have different assumptions. They assume different background mutation rate. They assume different background, genomic structure variation. This kind of thing it was considered in the model. That will be different. So it's hard to say, okay, this way you do the mutation call is better than that. It's but there are some tools that was very widely used. We can actually start with that. Like GATK, like SAM tools. So that's definitely the way to go.
So if I can add something from like a wet lab perspective. So when I was at MSK, we ended up doing a lot of validation of mutations, particularly for like exomes and genomes, because no matter which color you're using, it turns out that some of these things are either going to be artifactual or they're going to be
the system, biased sometimes.
Some kind of bias. So I think because of the expense attached, nobody has done a very comprehensive study. But in reality, the comprehensive study will require some validation. So you get a list that you get, let's say, mutex, somatic sniper, something like that. And then you capture those regions separately with probes, and you sequence it again at like a higher depth. And you run them, and you actually confirm which mutations were, called by one method or the other. And this way, you may gain some idea as to which color is going to be better. But like you say, one may be better in one context of tumor, particularly for cancer genomics. If there's a lot of rearrangements in copy numbers, and tumors that are kind of like more flat, you might get better calling with different one. But
right.
I I think ultimately you still need to validate, you know, some of these mutations if you're.
Yeah, exactly. It's a kind of like a process that you know, back and forth to interact with the lab, with here it's with you to discuss the things we discovered, what kind of characteristics this set has, you know, what we are missing, and then you know, have some maybe improve some new ways to target those things that we missed. So that absolutely is the, you know, the process and the way to deal with this kind of thing. The computational will solve is the part of the answer, but it needs to be constantly, you know, to modify or adjust the configuration, or, you know, to change the assumptions or something like that. Yeah. Okay. Thank you.